Hello, my name is Vikram,
My name is Ryan.
And my name is Nathan.
and this is our protein synthesis model. Transcription. RNA transcription works from the 3 end of the DNA to the 5 end of the DNA, or in the 5 end to the 3 end direction of the RNA. In our case, the 5 end is on the left, and the 3 end is on the right. This process is done in the nucleus of the cell by RNA polymerase. This process must occur as DNA cannot be directly transcribed into proteins, only RNA can. Thus, if you want to make new proteins, you need transcription to occur. Transcription is broken down into 3 steps: initiation, elongation, and termination. In initiation, after the RNA polymerase binds to the promoter, the DNA strands unwind and the polymerase initiates RNA synthesis at the start point on the template strand. In elongation, the polymerase moves downstream, unwinding the DNA and elongating the RNA transcript 5' to 3'. Afterwards, the DNA strand reforms a double helix. In termination, the RNA transcript is released and the polymerase detaches from the DNA. So over here, you can see that the top is the coding strand from the 5' to the 3' and then the bottom is the template strand from the 3' to the 5'. And over here is what the template strand would look like on the bottom, where there is a promoter on the very beginning, which is where the DNA polymerase will bind. And it will look for, the DNA polymerase will look for something called the Tata box, which is 25 bases upstream from where it starts to be transcribed from in a certain sequence. And then at the end, we have the termination sequence where it tells the whole process to stop. Over here, we have RNA polymerase, obviously attached to the promoter area. And as you can see, the DNA polymerase, it kind of separates the 2 strands, the coding strand and the template strand. And over here, we have transcription factors, which are essentially enzymes that help out throughout the whole process. And we have that RNA transcript being formed through the RNA polymerase.
So the 5' is synthesized first, receiving a GCAP, which is a modified form of the guanine nucleotide added onto the 5' after transcription of the first 20 to 40 nucleotides. On the other end, an enzyme adds around 50 to 250 more adenine nucleotides, which forms a poly-A tail. This process, which occurs in the nucleus of eukaryotic cells, facilitates the export of mRNA from the nucleus and protects it from being degraded by hydrolytic enzymes.
RNA splicing, the process by which introns are removed from the pre-mRNA and exons are stitched together, occurs in the nucleus of eukaryotes and does not occur in prokaryotes, as transcription and translation can occur simultaneously. RNA splicing is important because the introns that are removed do not actually contain information pertaining to the specific amino acids of the proteins. If they were transcribed into mRNA, the resulting polypeptide would be different than the one needed by the cell. RNA splicing is done by spliceosomes, which serves as a complex to catalyze RNA splicing. And as we can see here, the spliceosomes recognize the intron sequence AAAUGC, and as such, they excise it from the sequence, and then they stitch the exons together to make our mRNA to be sent off to the ribosomes.
Transcription is read through the mRNA strand sequence in the 5' to the 3' direction. Thus, our sequence of amino acids would be methionine, leucine, trypsin, and proline. This whole process of translation occurs out in the cytoplasm via ribosomes. So over here, we can see we have the 4 tRNAs that are attached based on the anticodons and the codons with the base pair rules. And then we have the release factor over here, and then this is the ribosome.
Initiation. In initiation, a small ribosomal subunit binds to an mRNA molecule. The initiator tRNA, which carries methionine, has an anticodon of UAC. It base pairs to the start codon AUG, which signals the start of the translation. The initiation factor brings together all the parts, and the hydrolysis of a GTP molecule provides energy for the assembly of the large ribosomal subunit. The initiator tRNA can be found in the P site, while the A site is free for tRNAs with the next amino acids, and the E site is the exit for the tRNAs.
Elongation occurs in ribosomes floating in the cytoplasm or ribosomes attached to the rough endoplasmic reticulum. The main difference between the proteins formed is that those formed in the cytoplasm usually stay and function in the cytoplasm, and those formed in the endoplasmic reticulum are usually part of the endomembrane system or are excreted from the cell, like hormones. A ribosome, after it's assembled, reads from the 5 to 3 direction. It finds the matching tRNA with the correct anticodon to bind to the A site, requiring GTP to form the bond. The nucleic acid carried by the newly arrived tRNA forms a polypeptide bond with the existing peptide chain already there. From there, the tRNA on the P side exits the ribosome through the E site, and the tRNA on the A site moves over to the P site, which also requires GTP. Of course, all these reactions are being assisted by proteins called elongation factors. And we can see just that shown in the image.
Termination. When a ribosome reaches a stop codon on the mRNA, the A site of the ribosome accepts a release factor, which promotes the hydrolysis of the bond between the tRNA and the P site and the last amino acid of the polypeptide, freeing the polypeptide. After 2 or more molecules of GTP hydrolyze, the parts of the translation assembly dissociate.
A silent mutation usually occurs on the 3rd base of a codon, and as the name suggests, it is silent and does not change the amino acid sequence, as shown. As the mutation changes, no amino acid is changed, so the protein function is preserved. So over here we can see that we have the 3 amino acids, and guanine was changed to adenine, but they both give out proline.
So a missense mutation occurs when a nucleotide on the amino acid sequence is changed, resulting in a different amino acid. In this situation, histidine was created instead of leucine, and the histidine is a positively charged ionic amino acid, whereas leucine is a non-polar amino acid. This replacement could remove some hydrophobic interactions or create ionic bonds where there previously was none. Changing the shape of the protein and reducing its capability to do its function.
With the deletion mutation, it shifts the so-called reading frame of the mRNA, which affects all of the nucleic acids downstream of the mutation, whereas in the substitution mutation, only one amino acid is affected. This can have a massive impact, such as in our example, where 3 of the amino acids are changed, going from a sequence of methionine, leucine, tyrosine, proline, to methionine, phenylalanine, threonine, and valine, which massively changes the structure, as phenylalanine has a massively different structure from leucine, which would change the shape of the final polypeptide and render the protein it forms potentially useless.